they're all related. So we looked at secondary structure prediction, which tries to predict whether a residue is in alpha helix, beta sheet, or term. And we also saw the dihedral angles, which in a way can also describe the secondary structure states of amino acids. And this Ramachandran plot is a plot of dihedral angle distributions in a protein, or you can also use it to plot distributions across multiple proteins. This is also sometimes used to as a quality control plot for protein structures, because you don't want to see a lot of things here. This area is a high energy area because it brings side chains into a clashing position, so it's usually avoided. If a protein structure has a lot of amino acids that fall in here, you usually look at that protein structure again. It might contain some errors. All right, so the tertiary structure refers to the exact coordinate of atoms. And there are also some supersecondary structures which take our secondary structure segments and sort of arrange them in multiples. And these also describe the fold of proteins. It's estimated that in nature we have around 10,000 folds. So any given protein, any given natural protein will fold into one of these 10,000 folds. Okay. So the hope in some of the structural determination experiments is that if we get enough, if we experimentally determine enough proteins, then we are likely to sort of have a complete sampling of these different folds. And once we have a representation from each fold, it should be easier to predict the tertiary structure of proteins. Okay. So given a protein sequence, amino acid sequence, tertiary structure prediction is taking that amino acid sequence and predicting the three-dimensional coordinates of these amino acids. So it's a more complicated prediction problem than just predicting whether it's an alpha helix or beta sheet. Okay. So you want to, in addition to predicting a local neighborhood, you also want to predict how the entire protein is going to fall. All right? And if you can take your protein and figure out which of these folds it will fall into, then you have a better chance of predicting the overall structure. Okay, so that's the idea behind the effort that goes into classifying these different folds. All right, some examples of these folds are beta alpha beta repeats. So you have a beta sheet, two beta strands separated by an alpha helix. Okay, a beta meander where the beta strands go like that. A Greek key, and this is in a real protein. So these are schematics, at two-dimensional schematics, and this is how they would look like in a three-dimensional figure. Okay. So you take this figure, and the Greek key structure is like that. Sometimes you may or may not be able to see the two-dimensional schematic within a three-dimensional figure. All right. So the 2D is just a simplification of how the three-dimensional configuration is. Some other examples are 4-helix bundle, where you have 4 helices making a bundle. Helix loop helix, where you have a helix making a loop and another helix. The coil structures they usually occur in transcription factors, where these arms are responsible for binding DNA or RNA. And these are usually leucine zippers, meaning you have leucine amino acids lining up the interface. And through hydrophobic interactions, they stabilize the two strands. The quaternary structure just refers to having multiple chains, having multiple proteins, multiple peptides forming a complex three-dimensional shape. And some examples are these homodimers of muscle creatine kinase. When you see a homodimer, it means that the two proteins are identical copies. So two of the same protein come together to form a dimer. The deoxyhemoglobin is a heterotetramer, and it's composed of two homodimers. So you have two hemoglobin genes, and they were evolved due to a gene duplication event. So two copies of alpha chains and two copies of beta chains come together to form a tetramer. All right. So the PDB, Protein Data Bank, has close to 100,000 protein structures now. And when you have a new structure, you want to usually know how similar it is to other protein structures. Similar to sequence alignment, we have structural alignment methods that will take two proteins and sort of superpose them onto each other. Okay. So if the two proteins, if their globular shape is highly similar, the problem is easier because you can figure out the corresponding residues easily. I believe this is a hemoglobin protein. Let me just check this again. It looks like one. And the structural alignment just geometrically takes two proteins and puts them on top of each other. All right, so the structural alignment problem is two problems in one. You usually want to do a residue-to-residue correspondence, just like you would do in sequence alignment. So you want to know if this is an alanine, which residue does this align with? Is it this one? Is it that one? Okay. So if you were to put the structures together, which residue would this A line up with? And it doesn't have to be an A, right? So long as they geometrically superpose on top of each other, you would consider that as a correspondence. Okay. The other problem deals with how distant the aligned residues are. So let's say these are the pair of residues that you align, that are corresponding from one protein to the other. This distance is an error measure that you would like to minimize. So you want to align the two structures in a way that they will superpose as best as they can, such that these distances are minimized, or some of these distances are minimized. Okay. So this is one alignment, this is another one, and this one looks like it's a better one. Okay. Because the residues are closer to each other in space. Since in most cases you won't get a zero error alignment, things will not align onto each other perfectly, you need to have an optimization method that will give you the best result possible. So structural alignment is a computationally difficult problem. It's difficult to simultaneously figure out which residues you should pair up and how they should superpose in three-dimensional space. All right. So because of that, most methods will give you approximations, and they will give you near optimal solutions. So this is the sequence. This looks like a sequence alignment where it's just corresponding the residues from one protein to the other, and you have a structure superposition that tries to bring these residues close to each other. Okay. But you don't know beforehand which residues to align. So the structural alignment problem both has to find the correspondences and also find the structural superposition that brings those residues close to each other. Right. So the error measure that's most commonly reported is the root mean square deviation. where you take the distance squared between corresponding residues, so that's just the Euclidean distance, you add them up and you divide that by the number of residues that you are aligning. All right, so this is an example. If this is the alignment, you just take these distances, square them up, and divide them by 5, and take the square root. It's given in the units of angstroms, because the structure is also given in angstroms. If you have an identical structures that are aligned perfectly, your RMSD will be 0 angstroms, right? Similar structures are usually in the order of 1 to 3 angstroms distance, and dissimilar structures have RMSD of greater than 0. So if you go back here, since you are free to choose which residues to align, you can get 0 RMSD by aligning 1 or 2 residues, right? So if you were to say, I'm not gonna align these residues, I'm just gonna align the 2 T's, then you can align them perfectly, right? So the RMSD will, in a way, depend on how many residues you are aligning together. So there's a trade-off between the coverage of your alignment, how many different residues you are aligning, and how accurately you are aligning them. So the structural alignment methods usually follow an iterative optimization procedure. They pick out an initial alignment, an initial set of correspondences. They align it geometrically. They align the 2 proteins geometrically, and then collect new pairs from that alignment, and then realign it after that. Okay. So let's just look at this example, which involves just the geometric transformation. So this proposition involves a translation and rotation operations. So you take this red protein and then you translate it on top of the gray one. And this is usually done by matching up the center of gravity, the center of mass of the 2 proteins. So once you do that, then the second operation is to rotate one protein so that the error is minimized. There's a method that will do this perfectly, in the sense that it will give you the correct answer. So it will give you a translation rotation matrix that best minimizes the RMSD error, given that you've already determined the pairs of amino acids that you want to align. STRUCTAL method is one of the older classic methods. So what it does is it aligns the 2 proteins, and you can use an initial alignment which can also be random. So you can randomly assign one residue from one protein to the other, to another residue from the other protein, and then you perform the geometric superposition. And then you would look at this geometric superposition, and you can recollect pairs that are closest to each other, and that gives you a new set of correspondences, right? So you started with this, you geometrically align the 2 proteins, and then you collect the new pairs, and then you got these new correspondences. And then you repeat the process, you geometrically align them, collect new pairs, geometrically align, collect new pairs, until things stop changing. So when your error converges, when the error does not improve anymore, or when your translation rotation matrix does not change anymore, then you can stop these iterations. So that's the idea behind structural alignment methods. There are other methods that do this differently, but usually this is the main idea. Okay, so let me go back to structure prediction. All right. So in your homework, you performed structure prediction using neural networks and also using statistics. And we compared that to a ground truth secondary structure. And that ground truth tells us what the secondary structure state of amino acids in our training set and test set are given their tertiary structure. So those proteins had known tertiary structures. Their structures are known in the protein data bank. Okay, so looking at their three-dimensional structure, we also know their secondary structures. Right? But going from tertiary structure to secondary structure is non-trivial, even though we have definitions as, where you have a helix and it makes hydrogen bonds and so on, but most proteins won't have an ideal shape for an helix. Okay, so things will be slightly bent. One amino acid may be out of place. So depending on what definition you use, you will get different answers for what secondary structure a residue has. So the DSSP is one of the classic programs that assigns secondary structures from tertiary structures. So this one usually looks at the hydrogen bonds formed between residues, and then it figures out which secondary structure state it should have. Other methods also use the alpha carbon atom distances and dihedral angles to define secondary structures. So I only mentioned 3 secondary structures, alpha helix, beta sheet, and terns, but there are different ways of defining other secondary structures. Some of them are included in alpha helices and others, but some are not. So there's a special 3 to 10 helix where there's a hydrogen bond between a residue I and the one 3 after that. So I plus third amino acid. There's a pi helix, which I believe is the more common one, where there's a hydrogen bond between I and I plus phi. Okay. And the beta turn is a special type of turn where you have a beta strand, and then this turn is usually named the beta turn. And the coil is sort of non-structured. Anything that doesn't fit into these categories is assigned to be a coil. You can also use the alpha carbon atom distances to define secondary structures. So you don't really need to look at hydrogen bonds. If you just look at the distances between alpha carbon atoms, that in a way serves as a proxy for whether those amino acids are forming hydrogen bonds. So these figures just plot the alpha carbon atom distances between I and I plus third amino acids, and then they look at whether it's a helix or a strand. So the distances that the helical structures form between I and I plus third amino acids has a different distribution than the ones that are in helices. Okay. So given this information, we can just look at these distances and be able to tell whether it's an alpha helix or beta sheet. And this program PALSSE uses that method. So depending on which method you use to define secondary structures, you will get a different answer. And as a result, you will get figures like this. So these A, B, C, and the 2 rows are two different proteins. The columns are different methods. So they just look at the tertiary structure and they assign letters to each residue. They assign which secondary structure state it has. And you'll get these different shapes. Sometimes this method gives a longer alpha helix, but this one gives a shorter one for the same tertiary structure. So for the same protein in PDB, these different methods will have different idea of where the secondary structure should be, or what secondary structure states there are on these proteins. Okay, so even though we predicted the secondary structure, you need to consider the accuracy that you get from the prediction in the context of the disagreement that these programs have. Okay? So depending on what you consider as ground truth, your accuracy will change. And you also know that you shouldn't expect 100% accuracy because things aren't really defined in a 100% agreement. All right. So you solve the problem of given a training data, where we know the secondary structures, find out what the secondary structures will be for a new sequence. There are multiple methods, and they give different results. The GOR methods use statistics. This PhD uses neural networks. Okay, and these red cylinders are alpha helices, and arrows show the beta sheets. So they mostly agree for alpha helices, but beta sheets are more difficult to predict. And the reason is that for beta sheets, you need non-local interactions. Okay? So that is a challenge for most prediction methods, because prediction methods usually use a local window. Right? So you use local information to make the prediction. So anything that is folding based on the local interactions, you will be able to predict more accurately. Right? But when you have beta strands coming together, it's sort of the information that you did not really capture in your encoding. So one strand could be at the beginning of the protein, and the other strand could be at the end of the protein. Since you did not encode those two parts in the same sample, you won't be able to predict it as well as the secondary structures that are more local. So determining success, Q3 and SOV are the most common measures, and SOV takes into account the segments rather than just the residues. So we looked at that. All right. So for some of the other prediction problems, there are more specialized scoring criteria. For the problem of predicting whether something is transmembrane helix and how it resides in the membrane. So you can take a protein and predict which portion of the protein is going to reside in the membrane, and which portions are going to be inside and outside the cell membrane. And for that, you sort of need a more accurate definition of a more accurate representation of where the transmembrane helix starts and where it ends. Okay? So this score takes into account that. It looks at where the transmembrane helix should start and how much the prediction disagrees from that. So it just looks at that. That's 2. This end is predicted with 4 error. The total error is 6. Okay. So now you no longer look at the letters here. So let's say this is transmembrane, 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 and the prediction is transmembrane. So you don't really care about these, how well you predict the residues to be transmembrane. You care more about how much the beginning and the end differs from the actual answer. All right, so we talked about that your training and test sets should be different, and that you should use a non-redundant or representative data set where things are different from each other. Otherwise, your learning method would memorize something, and you would think that it's performing well, but it's given a new protein that doesn't look like anything that you've used in your training, it may not perform so good. All right. So this is the statistical method that you implemented in the homework. So this is another evidence that the propensities or the secondary structure likelihoods are position dependent. Okay, so what that means is if you look at the position of the amino acid, so where that amino acid is, it will have a different likelihood for being a helix. All right? So it's not enough to look at whether an amino acid is alanine. It's also important to see where that amino acid is. Right? So this example shows that if you look at the alpha helical propensities of these 3 amino acids and plot the propensity, but now take into account the location in the alpha helix. Starting from the N terminus is 0 location, and this is the first residue, and so on. So whenever W is the first residue, its likelihood is that much. And then you plot them together, and then you repeat that for all 20 amino acids, and you plot them, and you get a graph like this. Okay? And you see sort of a repetition here. The hydrophobic residues like to be on the same side of the alpha helix. And that's probably because alpha, when they form bundles, you need sort of an interface where hydrophobic and hydrophobic partners will come together. Right, so the GOR method is another statistical method, but in addition to taking into account single residues, it also takes into account the neighbors. Similar to the way that you encoded windows in your neural network training, the GOR method looks at the neighbors and see how they affect the secondary structure state of a position. So even if you don't know what this amino acid is, all right, so even if I don't tell you what that amino acid is at this location, the fact that you have a proline here makes it less likely that this is an alpha helix. All right. So if you have a methionine, the 5 positions before that is more likely to be a helix compared to having a proline. Right? So there's an interaction between residues, and you can capture these statistics in addition to the single residue statistics to get a more accurate prediction. Okay. So that's the idea behind the GOR method. All right. Another commonly employed idea is to use homologous sequences. So if you are trying to find the secondary structure states of a protein, let's say this one, what you can do is look at all the other sequences that are homologous to this one. Okay? So you can do a blast search and get other sequences that are similar to this first one. And then you would do, let's say, prediction on all of these, and you can look at the consensus secondary structure. So the prediction will now involve, in addition to predicting your sequence of interest, you will also go get other sequences that are similar to it and make predictions on them also, and then see the consensus. All right. So the idea there is that if sequences are homologous, they should have the same protein structure. Okay. And even if your prediction method makes some errors, if you look at a bunch of similar sequences, those errors should, so in a way, cancel out. All right. And your prediction based on multiple sequences should be better than looking at just one of them. The nearest neighbor methods refer to the methods where, given a new sequence, you look at how similar it is to the ones that you already know and assign it the same secondary structure. And again, you would have a sliding window since you don't expect things to be identical in their entirety. You look at this window, you go look at your database, you can do a blast search, for instance, and find other similar subsequences. And your database will, of course, have their secondary structures already assigned. So your database will contain known PDB structures, known protein structures. And then you will combine these known secondary structures and get a consensus. Okay. So the one that has the highest frequency will be your prediction. So you combine the secondary structure states of similar sequences and make a prediction from them. All right, so the neural network method we covered. And I also said that there are some other specialized prediction problems, and these can be for super secondary structures such as coiled-coils, Here, in addition to predicting whether something is an alpha helix or whether something has this shape, you would also want to figure out how they are configured with each other. So what's the interface between the two coils? And again, there are some, you would first look at data that you have and try to find some statistics from it. And some of these statistics are that the ones that are on the interface are usually hydrophobic, and they are surrounded by charged amino acids. All right, so leucine zippers are usually in transcription factors, and the name comes from the fact that you have leucines lining up these interfaces, and they form hydrophobic interactions. A way to predict transmembrane helices is by just looking at how hydrophobic things are. Right? So membrane is a hydrophobic structure, and if you force a hydrophilic amino acid in there, it's going to have a high energy. It won't like being in the membrane. So the transmembrane regions of proteins will be more hydrophobic, and the things that are inside and outside the membrane are going to be more hydrophilic. If you take a sliding window and take the moving average of the hydrophobicity values of amino acids, you'll get a plot like this. All right. So if this is the hydrophobicity scale, you can easily sort of see which portions are stretches of highly hydrophobic amino acids, or stretches of hydrophobic amino acids. Those will be the transmembrane regions. Those will be the regions that are embedded in the membrane. But this doesn't tell you which side is inside, which side is outside. Right? So is this the inside? Is that the outside? There are some additional rules that you can use to orient your helix. Usually, the intracellular loops have a higher concentration of arginine and lysine, which are positive. Okay? So these rules are determined from known data. If you have a set of knowns, alpha transmembrane proteins, you can see whether they have anything in common. Okay? And you can do that either using statistics or using machine learning methods such as the neural networks that you use. Neural networks will usually don't give you rules like this, okay? Because it works as a black box. It will give you an answer, but it's more difficult to figure out why it gave you that answer. Whereas the simple statistical measures are more interpretable. Okay? So you can see what has a higher frequency. There are also hidden Markov models for transmembrane helix prediction, and we've encountered hidden Markov models in multiple sequence alignments, where a family of proteins can be summarized as a hidden Markov model. It's both the hidden Markov models can both evaluate whether a new protein is a member of that protein family, or it can also be used as a generative method and can generate new sequences. Okay. So if you need to generate samples from the same family, you can run through the hidden Markov model and generate representative sequences. So using consensus just refers to running multiple methods and see how they agree. So if you have a protein that you really care about, you shouldn't run just one prediction method. You should run multiple different methods and see how they agree with each other. All right. RNA secondary structure prediction is a slightly different problem. Its prediction usually involves finding out the energy of the fold and also the number of hydrogen bonds formed by the structure. So the RNA secondary structure will have these bulges and pairs of segments that are forming hydrogen bonds with each other. So the more hydrogen bonds you can form between A and U and G and C, the stronger the interaction will be and the more stable the RNA structure will be. So you can try different ways to fold the structure and see which one gives you the highest number of hydrogen bonds, under the constraint that an A can form hydrogen bonds only with AU and a G will form hydrogen bonds with SE. All right, so let me jump to, let's stop this for now.